I'm Sihan.
Hi, I'm Allison.
Hi, I'm Heijun.
I'm Sonia.
I'm Anthony.
And our experiment is measuring the elasticity of biopolymer gels. So what exactly is a biopolymer? So biopolymers are large molecules of repeating units linked together, and these are produced naturally by living organisms. And there are three main types. There's polysaccharides, which are sugars, polypeptides, which are proteins, and polynucleotides, which is represented by the image on the top right, which is DNA. And DNA is made up of nucleic acid monomers. And we have an image at the bottom that represents a polysaccharide or a sugar that is made up of repeating monosaccharide monomers. So these monomers are very important in creating this larger structures and larger networks of a molecule structure. And these structures are formed on the nanoscale, so 10 to the negative ninth. And so different arrangements of these structures can create different molecular shape and different molecular interactions.
Synthetic skin is a relatively new medical development, but is a rapidly growing subject. Our experiment pertained to two specific applications of synthetic skin, which were skin grafts and bandages. A skin graft is the replacement of healthy skin onto an area where the skin has been damaged, lost, or surgically removed. It can be used to treat skin infections, deep burns, large open wounds, ulcers on the skin, and skin cancer surgery, just to name a few. A bandage is a strip of material used to support or restrict the movement of the human body. Bandages are now starting to be engineered with biopolymers, as biopolymers have many useful medicinal and mechanical properties. For example, protein-based biopolymers increase cell proliferation, and certain biopolymers have adjustable permeability.
Okay, so currently in the market, we have a couple different products and procedures. So one product is hydrogel dressings and pads, and these are normally used to address burns, and these are made up of cross-linked polymers, which we will discuss later. Another product are bandages, like mentioned earlier, and these are just used for any cuts or wounds. However, for both hydrogel dressings and pads and bandages, they need to be replaced often and aren't able to stick onto the skin for long amounts of time. And artificial scaffolding or skin grafts use skin cells to help the body regenerate and heal. And scaffolding is basically nanofibers, so fibers on the 10 to the negative ninth scale, that use cell signaling to influence cell proliferation, differentiation, and biology. However, artificial scaffolding is pretty expensive, and it's mainly used for more extreme injuries. So currently, 3D printers are used to manufacture skin and muscles, and they use this by using biomaterials such as cells to create tissue-like structures by layering them on top of each other to imitate artificial skin. And Matroderm is a synthetic skin substitute that has recently been proven to be successful, and it was used with artificial scaffolding to improve flexibility and skin color. And this is made up of collagen and elastin, which are both biopolymers. And collagen helps facilitate new cell growth, and elastin allows tissue to resume their shape after being stretched. However, this is not available in the US currently, and it's also only used for more extensive extreme injuries.
So our research question is that how can we create a biopolymer bandage with skin-like properties that can promote healing over long periods of time? And our hypothesis is that out of gelatin, agarose, and cellulose, gelatin is the best biopolymer for creating a bandage with skin-like properties.
So the impact of our potential creation is that it could aid people with burns, cuts, and wounds. And it's more impactful than a normal Band-Aid because it can speed up the healing process, as that's a property of gelatin. It can encourage skin cell growth, which would cover the wound and make sure that it heals faster than it would normally. It can also act as a barrier to bacteria, as wounds often get infected, especially when they're deeply affecting the person, so it could prevent an infection.
So why do we choose gelatin, agarose, and cellulose for experiments? First reason is that there are three very common biopolymers. We can find them in the market easily. Second reason is that they are not toxic and have beneficial properties on human body. Like for example, gelatin contains healing properties and induces skin cell growth when applied. Agarose and cellulose are moisturizers and are compatible with the human body. The last reason is that all of them form gels that can be applied on wounds. And as you can see, the structure of gelatin is the picture on the top right, and agarose is in the middle, and the cellulose is the bottom one.
So one of the major mechanisms in this experiment is the mechanism of cross-linking. So cross-linking is basically when you form nanoscale connections between different polymers. So for example, like gelatin can cross-link itself. So gelatin can sort of overlap itself and form like a cross and then form bonds. So this makes the polymer stronger and more resistant to stress and high temperatures. So like in the top diagram above, like you see that blue line, that's the cross-link, and the two black lines, the wavy black lines, are the actual gelatin molecules, for example. And then the bottom is like the web link structure that actually forms like when you cross-link gelatin. So like, as you can see, like each of these points are different cross-linking areas, yeah. And an overview of our experiment. So what we did was we first synthesized these gels in Petri dishes. So we had to mix powdered gels in water, and then we had to melt them, make sure they form gels, and then cool them afterwards. So that took about a day. After that, we measured elasticity. So we cut out small rectangles and then measured them, measured how far they would stretch before they broke. So like that video over there.
During the experiment, we started with gelatin gels, and we were successful in creating a gel that was firm yet elastic. However, we weren't able to synthesize a cellulose gel. When we attempted to, we found that it was insoluble in water, so we tried to add a cross-linker, NaOH, and we also tried glutaric acid, and then that didn't work. So we tried dissolving the cellulose in another solvent that was nonpolar, but none of these alternatives worked. Agarose gel did form, however, we determined it to be too rigid at all tested concentrations to be considered a viable skin substitute in regards to elasticity. For our agarose gel, among all our trials, we found that a concentration of 3% for agarose was optimal and then remained constant. And then for our gelatin gel, among all our trials, we determined that a concentration of 50% would be optimal, and then we found that elasticity decreased as we increased concentration.
So in conclusion, what we found out is that the gelatin was the most elastic biopolymer of all the ones that we tested. And then this one right here, the 50%, that one was the most appropriate for bandage application because it was stretchy enough to be able to fit any wound if it's like a heel where it has to stretch like around or something like that. But it also has the appropriate thickness and sturdiness that it can last a long time. In the future, we hope to replicate other properties of skin, such as adhesive qualities, thickness, and absorption, so that it replicates skin as much as like physically possible because we want it to be comfortable. And then the results that we found suggested that a layered bandage would help improve efficiency a lot. So this is generally the plan that we were thinking about. So we would have gelatin as the base because of its healing properties, and then above it, we'd have a cellulose membrane. So we tried mixing cellulose with water and oil, but we think if we mixed it with a different substance that would help it become more of like a paste-ish liquid, then we could put it on the top, and that would be hydrophobic. So let's say you want to take a shower or go swimming. When you have this Band-Aid on, it wouldn't be a problem. Like normal Band-Aids, they just fall off, and they're not and this would not do that. But on the bottom, then would have a semi-permeable adhesive. So that would mean it would stick to the skin for a long period of time, and no water would get stuck under there. And then, of course, the skin on bottom that it would go on. Thank you guys for listening. Do you have any questions?
Hi, That was a great presentation. What was the process for forming the crosslinks, and then how did you know that they had actually formed?
So cross-linking, basically, what happens is like when we dissolve the gel and we heat it up, and we start cooling it, it starts cross-linking by itself. And basically, you can tell it cross-links because it forms a gel. That's pretty much the basic idea.
Thank you.
Great presentation. Did you do any studies with like times or light or like the degradation of the material? Were you guys able to do any of that analysis? Any known findings?
Well, we did have a short period of time, so we synthesized the gels, and by the time the gels formed, we weren't left with much time left. So our main concentration in this lab was focusing on how we could get the gels to form, and then testing their elasticity. So unfortunately, we didn't get a chance to try the degradation, but also these gels are pretty hardy, so it would probably take a lot of force. But that would definitely be a good future test.
When you talked about the layering of the Band-Aid, any thoughts of what type of material you could use to maybe incorporate a medication or within any of the layers?
Actually, we incorporated gelatin, which by itself increases cell proliferation. So that means that the gel, like, it promotes healing, like it promotes tissue growth. So that in itself would be like a medicinal property. Like,
Yeah.
we wouldn't need any additional, like, anything else to incorporate.
But yeah, the gelatin does encourage skin cell growth, but there is a possibility that depending on what substance was used in the bottom, you could incorporate that. That would be a good topic for future research with more time.
Okay, so I got to ask a few more sciencey questions. Okay, so the first question was asking about what made the crosslinks. So for every different kind of gel that you looked at, there's a different mechanism to make the crosslinks. But for this one that you focus mostly on the gelatin, can you tell people a little bit more about specifically how they crosslink, like using chemistry?
Okay, so gelatin is a protein or a polypeptide. So when they form cross-linking, basically the blue line over there, they form peptide bonds because they're proteins. So that's how they cross-link with each other. I don't know if that.
Do the peptide bonds form the crosslinks, or do they make the black lines? So we got the blue lines and the black lines. And the blue lines are actually many things linked together in a long chain. Sorry, the black lines are many things linked together. And then there's also the blue lines.
The blue lines are more like hydrogen bonds. So like the R groups of like all the amino acids, they interact with each other. So like depending on the R groups, they'll form like different bonds. So yeah, that's how they interact.
And that's exactly right. And then those bonds are weak bonds. And what's special about that in terms of how you process this? How did you use the fact that those bonds are weak bonds?
So because they're weak, we heated them up in water in order for those bonds to break at higher temperatures. And then once they cool down, those bonds would reform and then form a gel.
Yeah, that's exactly. And that's the really cool thing, is that you can manipulate them and break them and unbreak them, but they're unbroken at body temperature, but you can pretty easily assess where they are broken. So I just have one more follow-up question relating to the other person's comment about adding other medicines. So what is most of your gel made out of?
Gelatin.
What's the vast majority of it? Like by volume?
Water.
Water. So you got plenty of space to dissolve anything you want in all that water. And that's the super cool thing, is it's almost all water. You can put anything you want in there and it's still held. Good job.